right, it's my pleasure to introduce our next speaker, Divya Rose. She's a PhD student with the Mississippi State University's College of Veterinary Medicine, working with Dr. Matt Griffin at the Thad Cochran National Warmwater Aquaculture Center in Stoneville, Mississippi. Her research is centered on understanding the microbial dynamics in commercial catfish ponds, both between and within farms, as well as the influence of medicated feeds and pond treatments on the pond microbial community. Thank you for being here.
Thank you, Dr. Phelps. Good afternoon, all. I'm Divya Rose, graduate student from Mississippi State University. And I'll be presenting a part of my graduate research project, Pronounced Temporal Shifts Define Pond Microbiome Dynamics in Catfish Production Ponds. Just as a brief introduction, as we all know, catfish aquaculture is the largest finfish aquaculture sector in the country. Even though we have a lot of catfish microbiome research, it's largely focusing on host-associated microbiome. So when we think about fish and aquaculture products, they're constantly in contact with their surrounding water that can house different microbes. And characterizing these pond water microbiome will help us understanding the ecosystem function and health management in aquaculture. And my previous research or my previous objectives have shown us that even by keeping external variables such as host species, feed type, soil type, or climate constant, we see huge farm-to-farm variation in microbiome communities. These influence of farm-specific management practice on microbiome communities makes it difficult for us to do an inter-farm comparisons. And for that reason, the current study focuses on a single commercial aquaculture facility that is producing channel catfish fingerlings and are vaccinated with the live attenuated E. chloride vaccine. So my objective was to analyze microbial community dynamics in a commercial catfish production system throughout a production cycle. So first, I'll take you all through how we did our sample selection. Vaccinated with live attenuated E. chloride vaccine without knowing what was going to happen to these ponds, if they were going to break with any bacterial infection or if they were going to be treated a certain way. We went out to the farm and followed 32 different ponds throughout a production season. We collected pond water samples at 9 different time points, 2 samples per month from June through August, and 1 sample per month from September through November. And I want you all to think of time as an extension of other variation that we may see, which is associated with temperature difference, photo period, and increased feeding activity that we see in a commercial production system. So we had 14 ponds that matched our criteria of channel catfish fingerlings that were vaccinated. And luckily for us, we also had 7 ponds within that which were treated with fluorphenicol and 7 that were treated with Romed, which helped us make a comparison between what these 2 drugs will be doing to the microbiome community. And all these ponds were treated in the same month of September. So we collected the pond water samples, 50 mils from each corner of a pond, combined them, and was concentrated using centrifugation. DNA was extracted and sent off for amplicon sequencing to University of Minnesota. Just to give you all a timeline, the samples were collected in 2023 and were concentrated and stored back in a nucleic acid stabilization solution. DNA was extracted in 2024 within a month just to avoid variation brought in by long-term storage of nucleic acid. And it was sent out for sequencing this year. We received the sequencing results back 2 weeks ago, so we are still analyzing the data. I'll be presenting what we have so far. So moving on to my results, this is the relative abundance data of multiple bacterial family across this 118 water samples we had. I've divided them into groups based on the time points. We've collected these water samples just to make it clear. These are the time points when all these ponds were vaccinated. Just by looking at it, we can see that from the first half of June through first half of July, we see an increased presence of Cyanobacteria, and which decreases when we go further through the production cycle. And we see an increased presence of Microcystacea from July second half through September, as well as other families like Flavobacteriaceae, Microciliaceae, Nodocardiaceae. And we also have to keep in mind that this is the time when the temperature gets high. This is the time when we the farm has highest feeding activity, and also the time when we see bacterial disease outbreaks. So next we did the diversity analysis. First we did alpha diversity, which is the number of unique microbial taxa that is detected in each sample. So we were looking at how many different types of microbes did we find in a sample. But the way I'll be presenting my results will be in the form of first difference. It is like the rate of change of observed features or alpha diversity between two time points. So with the question we are asking here is like how different was taxa present in June when compared to July? And this is how the first differences of all our different sampling time points look like. And you can see that it's constantly changing. The alpha diversity is constantly changing. Just to bring in more sense to it, if the alpha diversity was not changing, our plot should have been a flat line through the global mean that we are seeing here. So the alpha diversity keeps changing throughout this production cycle. And from my metadata, I've also shown that we treated our ponds in September with two different drugs, and we wanted to see if it's changing the alpha diversity of these pond samples. And as you can see, in September, we do not see any difference. But when it comes to October and November, we see that the drugs are selecting for two separate groups of bacterial taxa, but it's not changing. The rate of change is not significantly different. We also looked at beta diversity using the Jacquard distance metrics. Because it gets noisy, I've not included the significance value, but you can see that the samples are clustering together according to each sampling time point. From previous results, from previous experiments that we've done, we've seen that the ponds have huge pond-to-pond variation, but the temporal effects are masking these pond-to-pond variation, clustering them together for us to actually see a shift in microbiome community throughout a production cycle. We can look at this in a different way, which is first distance. When first differences looked at how different the taxa in June was to July, here we are asking the question of how much did the overall pond microbiome community change from June to July. And again, as you can see, the pond microbiome community is continuously shifting throughout this production cycle. And again, if it was not shifting and it was stable, we should have seen a flat line here. So another way for us to, another analysis that we did was to look at the significant taxa that is contributing to these shifts when compared to my baseline data. So we used a program called linear models for differential abundance in microbiomes, which is designed exactly for studies like this, where we are doing longitudinal sampling of the same samples again and again. And this is the model that we used. We took time as the main factor of interest, and ponds were used as the random effect, keeping in mind that each pond is different to each other, but also is being repeatedly sampled over time. And the data type we used was row count data from 16S sequencing. So before getting into my results, we are visualizing the results from Linda using volcano plots. The way we look at it is I'm comparing each of my time points, each of my sampling time points to June 1, the first half of June, which I'm keeping as my baseline data, because this is before all the production related activities are happening in the farm. So every time point is compared to the first half of June. And on this plot, everything to the right of 0 shows an increased abundance of that particular taxa. Everything to the left shows a decreased abundance. And higher you go on the y-axis shows a higher significance. So we compared each time point to our baseline data, which is the first half of June, but I won't be talking about all these time points because of time constraint. We'll be focusing on these 4 time points when the active production is happening, and also the time points before and after these two. So let's start with first half of June compared to first half of July, and we see all these different taxa that is increased in abundance and those different taxa that are decreased in abundance. And when we get to second half of July, we start seeing a family that is of interest to us, which is Microcystaceae, which is consistently increased across during the next sampling time points, because it's there in the first half of August, second half of August, August, and during September. But however, when we get to October, we do not see a significant increase in the presence of family microcystaceae. And the reason we are interested in this one particular family out of all these other significant taxa is because we've had a previous study done by master student Alison Merchant, advised by Dr. Larry Hansen, that showed microcystinella toxin can increase mortality associated with bacterial infections. And we thought this would be a cool family to explore further. And all this time, I've been talking about a commercial setting, and the question that we have in mind is, okay, this is a farm. This is a farm system which is not set for a research experiment where it's not controlled. They are bringing in different production-related activities to these ponds. So these temporal changes that we are changing might be associated with all these different management strategies that they're putting in a commercial farm. But when we did similar study in a research pond in a much controlled setting where we are not feeding the fish too much, but just feeding them till satiation, we still saw these temporal changes, these temporal shifts, which were masking the pond to pond variation. So some of the future studies that we are interested in is to look at real-time PCR to detect Edwardsia Lake telluri in pond water samples, because all the 14 ponds that we've selected had E. telluri outbreaks according to the Aquatic Research and Diagnostic Laboratory reports. Another thing we are interested is to use the real-time PCR to detect toxic microcystaceous species in pond water samples. A 2020 study has done this in farm-based ponds, and it would be cool to see if we can detect this in our aquaculture ponds. We'll also look at the change in microbiome due to treatment and time treatment interaction after September. So to conclude, time plays a bigger role in microbiome community shifts than other factors, and changes in antibiotic treatment is not significant, or even if they are present, they are usually masked by these temporal effects that we are seeing. So key takeaways: microbial communities shifts rapidly, even every 2 weeks, as we saw from our first differences, and each pond behaves unpredictably. No two dynamics are the same. From this plot that you're seeing, you can see that each pond is shifting unpredictably, and it's so noisy. So for doing proper microbial comparisons, we need more dense and high resolution sampling. And as Matt would say, in hindsight, we should have sampled every other week to track and manage these stochastic changes. With that, I thank all these institutions and my committee members, and I'll take any questions.
Great job. Divya, that's some big data sets, complicated ones that you managed to present very well. Folks, we have time for a quick question. Feel free to drop it into the chat or unmute yourself. Okay, and if not, thank you very much.
Thank you.